Hey everyone! Hey everyone, this is Aurora with Supercharged Science, and today we are going to be doing a brand new class just in biology. And I wanted to test it out and see what you think. So if you like this and you want more, you want to go to my website, www.superchargedscience.com, and there are more lessons and in biology and life science that you can do. But the purpose for today's class is actually I've been putting together with a friend of mine who's actually a research PhD research guy, and we actually went to grade school together, and we recently reconnected, and he says, yes, I'm a biologist. I have a passion for teaching kids. And I said, wow, this is fantastic. So he's been helping me design a brand new class in cell biology because that's what his research is in. And so I wanted to test out a segment of that class with you right now. So I wanted to get your feedback on it. So in the comment section, you can leave me notes and what you like, what you want to see more of. We're just going to do a couple of just maybe 15-20 minutes of this introductory class. So if your kids have never done biology or if they've never done life science on this scale before, this is a perfect fit for them. Okay, so again, my name's Aurora with Supercharged Science, and I actually have 5 more science and biology lessons that you can do after this class is over. To download those, you want to click on that link that's right next to the description for this class, and it's superchargedscience.com/bio, B-I-O dash SAVS, S-A-V-S. SAVS stands for Science, Activity, and Video Series. So I put together a bundle of 5 different lessons that you can do, including extracting DNA and measuring the size of a molecule and doing microscopy and so much more. So you ready to get started? Okay, so let me share with you my slides right now, and let's get started. Okay, so one of the first things we're going to cover in this much larger course when it's ready is the difference between the different types of biologists. And there are quite a few areas of biology. You know, biology is the science that studies all living organisms, and that's huge. That's a huge field of study. Most students that study biology in college, they don't just study biology, they focus on one particular area, and a lot of these areas do overlap. So in the course that I'm designing, we're going to include zoology, biochemistry, molecular biology, ecology, botany, astrobiology, and more. And we have a whole other section dedicated to just marine biology and oceanography, so we're not going to cover that part in this new class. So it's a really big area of science, and it's different than most biology courses because I could spend all day going through every living thing and all the details about what it is and what it's not, and the details on the habitat. And there's a lot, and honestly, it's too much information. And a lot of textbook biology deals with that. And I realize I've been looking at the wrong camera the entire time, so sorry about that. There you are. I'm actually using a brand new camera today. So it's too much detail. And it's too much information to squeeze into our small time together. A lot of textbooks will do that. And honestly, it deals with a lot of memorizing about the inside anatomy of animals and the processes. But instead, you can find that stuff in a reference book anytime you need it. If you're studying jaguars in some small village somewhere, you're going to go find that information based on its geographic area, the type of habitat, and and what time period you're looking at. So that's not what we're going to focus on. This course instead is going to hit the highlights about what real biologists do in the field with their everyday lives. And we're going to study the big concepts that are in biology, and not just biology, but I'm going to show you how it connects with chemistry and physics as well. And so if you find there's something that you want to know more about after this course is over, after it's published, you can make a note about that and take a look at it after class and do further research, because that's what real scientists do. Okay, so why would people study biology? Well, before we introduce the classes, before we start talking about class, you have to cover the what is it and why do I care? So here's a couple of highlights I thought would be interesting. So cellular biologists, they study cells, right, including their function, systems, structures, how they interact. And so like molecular biologists, these biologists will usually work in the medical fields, and they're focused on the treatment of disease. Many of them will work as microbiologists, as biochemists, or something called biophysicists. So if you get a career in cellular biology, it'll usually take you about 4 years to get a bachelor's degree, and then you'll have postgraduate work like a master's degree. And some of the things you'll study include chemistry, biology, physiology, and laboratory research. Now, these types of biologists use several different sciences together, combined every day. They combine genetics, biochemistry, cell biology, pathology, microbiology, physiology, tons of ologies, right? They get to do lab work. They get to participate in experimental trials with animals. They get with animals or humans to help find cures to diseases or help making the healthy body perform even better. So a microbiologist, molecular biologist, are really good at what they do because they have to study so many different areas in order to do their job. They might spend the day looking at DNA strands, finding cures for diseases, or figuring out how to reverse the damaging effects of pollution, just to name a few. So if you like solving puzzles and doing experiments, this might be the area of science for you. And biochemists tend to do the same thing, but they focus more on the chemistry side of things. But both of them could really do the same job, and the jobs pay really well too. For example, molecular biologists were the ones that found organisms that seemed to be a cure for a certain type of cancer, but it only exists like in the puddle. They found it in one puddle in a deep dark cave in the one area of the world so far. So their job is to figure out how to duplicate that and make it more sustainable and make it scalable on a much larger scale so they can help many more people. And so they're going to study things like the chemistry of living things, cell development, growth, hereditary disease, and that sort of thing. So there's a lot of different areas. Okay, let me show you my other slides here. And so we're going to skip over to, I thought you would be interested in the different types of microscopes that these biologists use. So here we go. Now, by the way, if you've just joined us, my name's Aurora. This is Supercharged Science. We are doing a brand new class. This is just a portion of the class that I'm designing and developing. It's not quite done yet. It's about 2/3 the way there. And we're looking for feedback. So I'm going to be presenting some things and want your feedback on them. Let me know what you'd like to see more of or what you really like. And that way we can include it when it's ready. It should be ready very soon. Okay, so we're going to take a look at the different kinds of telescopes, different kinds of microscopes that biologists will tend to use in the field. Now, microscopes come in a couple of different types. You probably are familiar with the optical telescopes that use light and lenses to magnify an image, right? You take a pair of binoculars and you look through them backwards, and now you've got a microscope. So I'm going to teach you in this course how you can actually use a laser microscope. It's a lot like using a water lens. It's super cool, and you get to see what's swimming around inside the water that you can drink, for example. So you can have all kinds of different kinds of microscopes. You can have laser microscopes. Those are designed for things like thick samples with different focal distances. And so if you look at the difference here, the bright field image is taken from a regular compound microscope that you might have at a local high school. The image on the right is taken from a phase contrast microscope. You can see much more detail on the right side. Do you see that? Here. So bright field and phase contrast right in here. Okay. So before I show you the next microscope, let's talk about lasers just for a second. Now, lasers are really useful, right? We use them every day. We use them in scanners. We use them to measure things. We use them in CD players, the Blu-ray DVD devices. They're like everywhere these days, right? Okay. So if you take well, here, let me just show you a picture because this is a really cool idea. If you take a green fluorescent protein from an animal like a jellyfish, this tiny part is so small you can't even see it, and you take it specifically from its DNA, you can actually create light inside of another organism when you shine a special kind of light on it. So scientists are using this idea to create fluorescent tags to track things like proteins so they can watch cancer cells or viruses multiply and spread while they're still inside the body without actually doing anything else. So you can insert this fluorescent protein, this tiny part of a cell, inside an area and it'll light up and you can watch it in real time. And that's one of the big reasons that biologists study animals. It's so they can understand human health. And it also turns out that you can take this special protein and use it as an optical amplifier for green laser light. So basically, the cell itself becomes part of the laser. Scientists pulse different kinds of light through a cell, through a jellyfish protein, and for the most part, the jellyfish like just glowed. But at one particular wavelength, the light came out all one color, pure green, and increased in brightness and was focused and not all diffuse like a regular light bulb. So basically, they were using a living cell to amplify the light inside of a laser, which is a really unusual way to make a laser. So the animal itself became the laser. Now, there's like no application for this anywhere. It was just kind of a surprise when they were doing their research that, wow, that happened. Okay, let's go back to how microscopes are really used. Okay, you ready? Okay, so in this next part, there's a really cool new kind of microscope that's are fluorescent, and they're called fluorescent optical microscopes. It allows you to see the thing you're looking at alive, and it also allows you to stain it in a way that you can see it, which parts are which, even if the parts of the organism are alive or dead at the time you're looking at it. So you can use it to be able to see different things inside. So what you're looking at, oh, did that not play? There we go. What you're looking at here, down here, this is the red blood cells in a zebrafish, and you're looking inside. Oh, now this is a fruit fly that's actually developing. Do you see how you can see different parts of it very clearly? Okay, now I think this is a zebrafish that's developing. So depending on what you want to look at, you can focus your area of study on one particular area. So this particular kind of microscope is called a fluorescent microscope. It's a fluorescent optical microscope. Okay, so you see the different organisms over here. We have a paramecium, we have a virus, and we have an ant. Now, the way you're going to see different things, you're going to need different types of microscopes. So the paramecium in the upper right, this is an image taken from a regular old microscope you'll find at a high school. This is an optical microscope image. Now, if you've got an electron microscope, then you actually have two different choices of which type of electron microscope you are going to be using. An electron microscope basically uses magnets that move and focus the electrons and focus the image or bounce it off the image. TEM stands for Transmission Electron Microscope, and it uses electrons to get a really high magnification and resolution in 2D. Now, a scanning microscope, so an SEM, scanning electron microscope, uses 3D black and white images on a computer screen. So these two images here, so we have 2D and 3D, so this would be your scanning electron microscope image, and this is your TEM, transmission electron microscope. Okay, the problem with electron microscopes, other than being like really expensive, is that the specimens are no longer alive when you are taken because of the way the microscope works in the first place. For a scanning microscope, you have to put this thin layer of metal on it in order to get the electrons to bounce off of it. So it's good, but it's got its limitations, especially if you want to study what organisms do when they interact with their environment and with other organisms. And an electron microscope's not going to let you do that. So there are special laser microscopes. There are multi-photon excitation microscopes, say that 5 times fast, that use lasers to make high-resolution images to observe like nerve cells and blood flow in the brain. Now, what you're looking at in the video, the colors, now there's people talking about it, but the colors are there because they've used a colored protein so they can observe it in real time. Now, the image in the upper right, that's a mouse intestine using a titanium sapphire laser. The green cell is the nuclei and the blue is the mucus of the, anyway, it's just different parts. They stain different parts so they would show up. Now, titanium sapphire lasers are tunable lasers. Those are lasers that can change the frequency of and the color of light that comes out the end. So it's a tunable laser. And these ones emit red light and near infrared. Now, these types of microscopes, they've been around since about 1982. And you can see the image, the image here on the upper left, that is a microscope that took the image in the lower right. So that's what the microscope actually looks like. Isn't that cool? Yeah, it makes you want to be a scientist, huh? Okay, so let me pause here for just a minute and see if we have any questions that have come in. Whoop, no questions yet. So if you have any questions or if you like what you see and you want to see more, let us know. I'm just giving you kind of a brief overview of or just little snippets of a new class that we're developing here. If you just joined us, my name's Aurora with Supercharged Science, and we are doing a snippet class, kind of like a sample saying, hey, do you like this? Would you want to see the whole course in it? Of a new biology series that I am creating. And let's see, I could probably do a little bit more. Let me pick a couple of things here that we can talk about. What would be good? Hang on. So let's see. Oh, we have basics of biology. Let's see if we can get something interesting here. Oh, okay, so that's our experiment. Oh, okay, so we can talk about, well, this is kind of weird to talk about. We didn't actually talk about this much, but so this is showing mitosis. Mitosis is exact copies of cells. When you form a scab, like if you get if you get a cut or you're riding your bike and you get, skinned your knee, you'll have a scab, and that's actually mitosis happening. When a lizard loses a tail, that's mitosis. You start with one cell and you wind up with like zillions of cells that are exact copies of the original. That's why lizards can regrow their tails and your scabs eventually heal, right? So mitosis is how a cell makes more of itself. It's how living things repair the damage and how they grow and just maintain their condition. Now we're going to do an experiment in a minute in this course that I'll show you just what the experiment is, that we're going to figure out how much time it takes for a cell to go through each stage of mitosis. And in the video you've seen here with the fluorescent proteins, we can see the red chromosomes and the green little spindles that are attached. Now, mitosis doesn't move in jumpy steps. It's kind of like a movie where everything just flows from one thing to the next when it's ready. And so what you're seeing here is a process where the cell is actually splitting to become two cells, each of which has the same number as the original cells, the same number of chromosomes. And they keep dividing until a protein tells them, okay, stop, you're done dividing now. So let's see, in the last step here, let's see, and then we talk about the different steps here. We don't have to talk about that now, but so basically this is what you're seeing here. This is an animal cell. Plants do something a little different in the end than animals do. So the experiment that we do, imagine, this is an experiment you can do at home actually. So here, pay attention for a second. Okay, so imagine you've got an onion in your hand and you're going to hold one of those little stringy white hairs that comes out of the bottom. So now imagine you're going to take a knife and you're going to slice through that thin piece and stick it under a microscope. Okay, so I'm actually going to do these steps for you. I'm just pretend like that's what you're doing. Okay, so now we're going to add a little blue stain so the cell structure shows up. Okay, so that's what this is here. This is a root tip, and we've taken one of these little hairs and we sliced it and we stuck it in our microscope and we stained it so we can see it, because otherwise it'd be near invisible. Okay. Okay, now we're going to adjust the lens until each individual cell becomes visible. So we're going to zoom in on this area. See all these little dots? They actually look like this. Okay, so we're focusing on the tip of the onion root. Okay, so just zoom in just right above the end. Here's the actual tip, and so this is right above the end. Okay, so now what we do is we take a sample here of at least 30 different cells. Each one of these little things is a cell. Okay, so now we're going to count the total number of cells, and how many do we see? Okay, so then you count them up and add them up. There's a lot, right? Okay, so now what we're going to do, after we've counted them up, we're going to go back and we're going to count which ones are in which phase. Okay, because you see like this one looks different than this one, right? This one looks different than that one. But, some of these look really similar, right? These are looking similar here. This one looks totally different. I don't see any more of these. But you can kind of, even without knowing what the 4 phases are, you can kind of see similarities, things happening. You see little dots appearing here. You see no dots in there, or it might just be fuzzy. So you're going to find which one of the 4 phases, and the phases are metaphase, prophase, anaphase, and telophase. And so we're going to find out which one is which in here. And so I can just show you, because we kind of skipped over this part in the course. But so we've got the interface, we've got all the different, all the steps, the 10 there are 4 main steps, but we can break those down into 10 distinct steps. And what you do is you count up how many are in each one, and then you'll be able to tell the cell spends most of its time in which one of these. Now, I'm not going to tell you the answer because I want you to figure it out, but if we go back to our original sample, this is what it looks like when it's zoomed in. Oh, it looks like I am telling you the answer. Whoops. So we counted up 403 cells here, and it looks like if you just take a quick look, where does the cell spend the most of its time? Yeah, in the interphase, right? And the least amount of its time in the anaphase and telophase. So you'll see very few of those in here. So based on that data, so this is us doing a scientific experiment with a lab work here, just like a real scientist, and crunching numbers just like a real scientist, you can see that the onion root spends the majority of its time in interphase because that's the phase that most cells are in. Okay, prophase is the second longest stage. This tells you that the onion spends most of its time getting ready for mitosis, which means that the organelles are doubling, the DNA is replicating, protein synthesis is happening, all these other steps are happening because that's where it's spending most of its time. And cell division, if you notice, doesn't take very long, right? Because these last 2 phases, when the cell is actually dividing, it happens really fast. It's kind of like getting ready to take a test, right? You have to be really sure you know everything, and then you spend like days and weeks preparing and planning. But how long does the actual how long does it actually take to take the test? Yeah, it's quick and easy if you're prepared, right? Same thing with cell division. It's the process of getting ready for this part that usually is the longest, and that it takes by itself. Okay, great. Well, that's probably, here. That's probably plenty. Let me pause for a minute here because we've been going for like 25 minutes. I just want to pause here and just make sure that you guys can send in your feedback and let us know what you're thinking. And if I can find the button so I can get my face back on the screen. There it is. Hello. And so let us know if you're enjoying this part. Would you like more of this? Because there is, I shared with you like 5 slides out of, or maybe 10 slides out of 120. So this is a huge new class that we've been working on, and it's exciting to share these things with you. And for example, I have I think 120 new videos in chemistry that I'm still trying to find time to post. But those are all completed and ready to go. So we have a lot of new content. If you like this content and you want access to like all of it, you want to go to www.superchargedscience.com/easy. Like, wow, that was so easy. And your kids can learn science just like this today, where we break it down into small steps. They're doing experiments, they're doing labs just like a real scientist. Now, if you like this lab and you want 5 more freebies in the downloads, you want to go to www.superchargedscience.com/bio, bio-SABS. I think the link is in the description. Just click on it. You'll be taken to my page where you can download that for free. Just enter in your email and tell me where to send it, and I will send that right to you. If you have feedback on this, I'd love to hear it. Put it in the comments so I can do more of what you want to see. We're a friend of mine and we had gone to grade school together, and now he's a PhD research scientist who has a passion for teaching kids, and his area is cellular biology. And so he's been helping me put this together because I'm much more into physics and chemistry. I kind of wasn't sure how to package something together, but he's actually been teaching kids for years, and we just recently reconnected last year. And so with any luck, I can get him to come on camera. He's kind of shy, but come on camera and at least introduce the labs to you that he's spent a long time working with me on. So it's very exciting to be collaborating with people. I hope you've enjoyed this. I'm going to pause here, and if you want to get the rest of it, go to www.superchargedscience.com/bio-SAVS. SAVS stands for Science Activity and Video Series. And you can download that for free. And we have all kinds of new classes coming. Tomorrow we're going to be doing another segment of a class, and we hope to see you tomorrow morning. I'll see you soon. Take care, everybody.